Polymerase chain reaction, also known as PCR, is used to copy or amplify particular DNA fragments. It starts with a sample of double-stranded DNA and a certain fragment of which the user is interested. So let's say I want this section here and I want to copy or amplify this certain region. The first step is the denaturation step, which sets the temperature to 96 degrees Celsius and separates the double strand into two single strands by breaking the hydrogen bonds that connect between the nucleotides of each base pair. These nucleotides are built from a sugar phosphate backbone and a nitrogenous base that make up a single unit of DNA. This is seen in figure 1.15. These two individual strands are then cooled down to 55 degrees Celsius with the addition of an external synthetic DNA oligonucleotides, which are known as primers. These primers are essentially an artificial product that can bind to your original DNA sample using complementary bases during the annealing stage. Now that these short primers have attached, we raise the temperature to 72 degrees, and here you can see this extended blue arrow of the primer. This is a result of the polymerase enzyme and added nucleotides. This specific polymerase enzyme is built for heat resistance and derived from a microorganism called Thermus aquatis, giving it the name Taq polymerase. This completes the first cycle of PCR, which doubles our interested part of the DNA sequence. This then creates a chain reaction where our DNA fragment doubles through every stage, as you can see here, just like how meiosis doubles from the initial parent cell to 2 haploid cells, then 4 haploid cells. The role of peer review and collaboration in PCR development began with Watson and Crick's peer-reviewed publication of the double helix model and base pairs within DNA, which was followed by Arthur Kornberg's team and their discovery of DNA polymerase, which was a key component in PCR development. This majorly contributed to Kary Mullis, who used a strong prerequisite of molecular biology built upon peer review and that highlighted issues in reproducibility throughout his collaborative development of PCR. This led to improvements in the methodology before publication within Methods of Enzymology in 1987, ultimately emphasizing the importance of peer review. Possibly the most important development came from international scientific collaboration and review that emphasized the initial labor-intensive PCR method of adding fresh DNA polymerase to each cycle, approximately 25 to 35 times. This is because high temperatures eradicated the polymerase after each denaturation. Following this, researchers at Cetis Corporation responded to peer-reviewed feedback with microbiologist collaboration that led to the discovery of Taq polymerase, which resisted these high temperatures and withstood many cycles. Now, a great example of mentioned collaboration and peer-reviewed networks includes the Human Genome Project throughout 1990 to 2003, which targeted the analysis and sequence of the entire human genome. This project significantly relied on PCR as a core foundation and connected several researchers that conducted a multitude of peer-reviewed articles on PCR-based sequencing methods, which enhanced standardization and credibility nationwide. Ultimately, this showcases the importance of publication and peer review in the development of scientific knowledge. PCR technology is one of the most revolutionary scientific advancements, and through its development and evolution, it provides a strong insight into the intertwined relationship between science and society. A key example of this includes the medical applications of PCR. This highlights the HIV crisis response between 1980 and 1990, which created a societal demand in PCR technology for diagnostic requirements and resulted in the rapid progression of HIV detecting methods, such as analyzing RNA blood samples through PCR. Furthermore, a more specific example includes the use of quantitative PCR to develop viral load testing, which contributed to HIV treatment, letting physicians analyze the efficacy and possible further development of such methods. Parallel to the surge of PCR development through the HIV crisis, cancer diagnostics also played a pivotal role in PCR-based techniques as it prompted the development of BRCA1 and 2 gene identification, which are genes associated with breast and ovarian cancer. This development illustrates the intersection between science and society as it highlights scientific capabilities and accelerated development by consolidating the societal issues of women's health and evolving the medical ethos. Ultimately, this exemplifies how nationwide collaboration advocated for societal necessities and how such necessities majorly corroborated with the expansion and progression of PCR in a medical scope. Whilst mentioning the medical application, PCR is also commonly used for forensic sciences that help reshape society's judiciary, institutions, systems, and practices. The appropriation of PCR-based DNA profiling within criminal justice cases serves as the best example of science and society's interconnected relationship. For example, PCR was used to amplify certain DNA blood samples linked to O.J. Simpson's case and formed evidence through genetic profiles. Essentially, this insinuates the direct link of science and society by evaluating the effectiveness of scientific methods and practical applications of legality.